Okay, I believe we got somewhere off here. We were looking at the breakdown of proteins. We were talking about the zymogen, which is that cocktail, all of the granules that have these non-activated proteases stored inside, and then how we release those granules. So here, how we release those granules in that pancreatic cocktail, that pancreatic juice, which has several different enzymes in it, how we release that from the pancreas via the pancreatic duct into the duodenum. We were talking about how we then activate those proteases. So again, in an attempt to prevent digestion of the pancreas while these things are stored in the pancreas itself, and as they're moving through the pancreatic duct, they're inactivated. So they're not in their active form, but as soon as they get into the lumen, the right conditions exist to where they can become activated. So we've got the aminopeptidase and enterokinase. These are the brush border enzymes. So these are enzymes that are along the cells, the enterocytes, the cells that line the duodenum, and they will then activate the first inactive form of trypsinogen to trypsin. And from there, trypsin will activate the others. So chymotrypsinogen can be activated to chymotrypsin, procarboxypeptidase, can be activated to carboxypeptidase. And then all three of those can start breaking down proteins into amino acids in order to get them absorbed across the epithelial membrane. Okay, so several different players here. These, as I mentioned earlier, these can cut peptides at different locations, different spots. They can be enteropeptidases, they can be exopeptidases, they can cleave the proteins in different mechanisms or different parts, depending on the size of the protein, the type of protein, and how. Remember, proteins are very complex in their structure. So there can be secondary structure, tertiary structure, quaternary structure. And so there's a whole host of different classification of enzymes to cut different structures and different types of proteins. Okay, moving on here to how we start absorbing those proteins. So kind of similar to what we saw in terms of the mechanisms here, but there is another option. So we can move amino acids into our epithelial cell and then across the basolateral membrane. So we get them into the cell by facilitated diffusion, and we get them across the basolateral membrane by facilitated diffusion as well. But we can also, to some extent, move smaller peptides, so dipeptides, tripeptides. We can move those into the cell by secondary active transport linked with sodium as well. So the similar mechanism that we see here for how glucose gets into the cell, we can do this for larger peptides. Again, smaller peptides. Dipeptides, tripeptides that are not as complex. They're only that single primary structure of the protein, still not as complex as some other larger proteins. And so we can get those into the cell by secondary active transport. But once they're inside the cell, they must be further broken down to amino acids. That way they can cross the basolateral surface of the cell by facilitated diffusion. Okay, so just to say that here again, we can get amino acids across the apical surface and the basolateral surface by facilitated diffusion. Or we can get smaller peptides, dipeptides, or tripeptides across the apical surface by secondary active transport with sodium, and then by facilitated diffusion as amino acids once they're broken down to amino acids across the basolateral surface. Okay, this is not the case, really important to contrast this to what we saw with glucose. Anyone remember what state carbohydrates had to be in order to be absorbed in any fashion across apical or basolateral surface? So what was the state that we needed our carbs to be in before they could be absorbed? Excellent, monosaccharides. So they must be their smallest monomers, must be monosaccharides. Here with proteins, we're seeing slightly more flexibility in that we can still have peptides move into the cell, but then they can further be broken down to amino acids in order to get into the blood. Okay. Moving on to lipids. So lipids are very unique. They're unique in their structure. And so they're also going to be unique in how they are digested and absorbed. Now, a lot of our diet is fats, about 50 grams per day of lipids. 90% of that is triglycerides. It's going to be that glycerol backbone with 3 fatty acid tails. And so they face a special problem in terms of moving around the body, being broken down, and also being absorbed into the circulation. So for one, they cannot enter the blood, right? They're just not lipid soluble. They cannot exist in our capillaries. And so we'll talk about the other circulatory pathway that they take in order to be absorbed. Also, they need help, right? They're really large, they're non-polar, they don't mix well with our other intestinal contents or stomach contents. And so they usually kind of form themselves into these large fat globules in the circulatory, in the GI tract. And so because they're so large, that poses a second problem. So not only are they non-polar in that they can't interact with aqueous environments, but then they form themselves because of that disaffiliation or unattraction to the aqueous environment. They kind of just lump together and form themselves into these large fat droplets that, again, pose another problem in terms of breaking them down to their smaller components. Okay, so we'll talk about how we can overcome those problems with the help of lipase and bile salts. So lipid digestion it takes place with the help of lipases, and lipases are part of that pancreatic cocktail, that pancreatic juice. So it's one of the enzymes that comes from the pancreas in the pancreatic duct and is dumped into the duodenum, and it can start breaking down fats. Now, again, because lipases can only act near the edge of the fat droplet, right? It can't go deep into that larger fat globule or fat droplet. And so again, because of that problem that's posed with this large fat droplet, we need bile salts to come in to break that larger fat droplet into smaller fat droplets. And this is a process called emulsification. So this is why bile is made in the liver, stored in the gallbladder, and then released into the duodenum, because only with the help of bile can we break down fats, can we start breaking down fats from those larger fat droplets into smaller droplets. And then lipases can come in, they're going to better be able to get all sides, all surfaces of that smaller fat droplet and start catabolizing that triglyceride into smaller components, fatty acids that can be absorbed across the epithelial cell. Okay, and then just the clinical connection here. This is why if you have your gallbladder removed or any type of gallbladder surgery, you are really advised to avoid fats in future, right? Because it's going to be really hard for your body to store the necessary amounts of bile. So your liver may still make bile, but just to have it in the right quantity to be able to attack those fat droplets in food is not as quick as a response as if you had that gallbladder that could readily supply bile. And so usually after gallbladder surgery, you are advised to have a low-fat diet and to minimize fat intake because of this reason. Okay. All right. Let's talk about how these bile salts work. So they are synthesized in the liver from cholesterol, also using that bilirubin that we spoke about yesterday. Remember, bilirubin is a waste product, but we can also conjugate some of that and convert it to this more useful form, which is this bile salt. It is secreted into the duodenum via the mechanisms we talked about before with the ampulla of Vater, joining with that pancreatic duct. And then it is amphipathic. So it has polar properties. It has polar groups, which we can see here. So it's got these blue polar groups. These parts can interact nicely in aqueous environments. And then it's got these hydrophobic or non-polar parts, which can interact better or more nicely with the fats themselves. And so in this way, it's going to be emulsifying the fat, right? It's not necessarily digesting it because bile is not an enzyme. It's kind of just mechanically breaking the fat apart. And so lipase, which is the enzymatic component, can come in and start catabolizing the fats into their monomers. Okay, so now lipase can come in. So let's look here at the schematic showing this larger fat globule, again, non-polar, not interacting well with the polar water or aqueous environment around it. Bile salts can come in, start mechanically shredding that larger globule into smaller globules by its amphipathic nature. And then now we have smaller, more manageable fat droplets instead of a fat globule, right? So kind of much smaller structure here. Then we can have lipases come in and start breaking this down into smaller components. So we can break it down from triglycerides into monoglycerides. We can pull that glycerol backbone off. We can have fatty acids that are separate. And then some fatty acids can be absorbed. They are monoglycerides. Others can be converted to micelles. And this is going to depend on how much fat there is to be absorbed, right? Again, this is how we regulate this. So we can convert them to micelles, and micelles are structures, we would have talked about them, I think, in Unit 1, where we have the fat in the middle and all of these phospholipid tails around it, kind of sectioning it off. And phospholipids have that polar head, so they can interact a little bit better with water. And so we can move these fats around better as micelles until they're ready to be absorbed. Or if we've got a lot of micelles, we can convert them in the opposite direction into fatty acids so that they can be absorbed. So depending on equilibrium here, how much of our substrate or product we have, that will drive the reaction either forward or backward in, an equilibrated fashion until we are able to absorb as many fatty acids as we need. Okay, so this happens under the influence of that lipase, which can catalyze this reaction forward to micelles or backward to fatty acids, depending again on how much fat is being absorbed. Okay. Let's talk about how we actually have that absorption taking place. So kind of going through this from the very beginning here. We have our smaller fat droplet. So this is what is left after we have broken this down from that larger fat globule. We've got triglycerides, which are the glycerol backbone. It's got 3 fatty acids. We also have phospholipids that are now being broken down by the lipase that's acting here. So now we have smaller monomers. We end up, after we've broken that down with lipase, we now end up with our monoglycerides. So these are fatty acids that can be absorbed directly into our epithelial cell. And here is that back and forth reaction. So lipase can catalyze if there's a lot of fatty acids and we just can't absorb them fast enough into the lacteal, then we're going to just shuttle them off into micelles. They can move around much easier. They can be transported until they're able to be absorbed as micelles. And then if we have a lot of micelles, we can shuttle them back to fatty acids where they can actually be absorbed, all right? So again, we're working to maintain equilibrium here with lipase catalyzing this reaction back and forth. Now, once they are fatty acids, we can bring them into our cell, and there are several steps before we can actually start moving them into the lacteal, into the lymphatic system. So essentially, they're going to be repackaged. So they're going to be repurposed back into triglycerides. So we're going to put them back together in the smooth endoplasmic reticulum. Then they're going to be shuttled off to the Golgi apparatus, right? These are all organelles inside the cell. The Golgi apparatus is like the post office, if you will. It's going to repackage them into a vesicle. They're going to be called a chylomicron at this point. And then as a chylomicron, again, they're better able to move around, packaged in this vesicle. They're going to be secreted by exocytosis. They're going to enter the lymphatic system via the lacteal. And the lacteal was that lymphatic duct, that green structure that we showed that is in the center of the villi, right, or the villus. I do want to mention here before I move on, so the fatty acids themselves can move right through the membrane. They don't need any energy. They don't need any transporters or carriers. They are fats. The membrane is fatty. It's fats in its nature. It's phospholipids. And so they can go right through this epithelial layer and into the cell by simple diffusion. Okay. Oops. All right, so let's kind of go over the steps that I just mentioned in terms of getting those fatty acids into our lacteal. So Once we have fatty acids, they cross the membrane by simple diffusion. They go to the smooth endoplasmic reticulum, which is this organelle here in purple. They are then reformed back into full triglycerides. So we're going to get 3 of them, package them together, and then from there, they are shuttled off to the Golgi apparatus. In the Golgi apparatus, they're going to get repackaged. They're going to look completely different because we're going to package them in a vesicle that is now called a chylomicron. Okay, once they are a chylomicron, again, they are able to move right through the membrane, no carriers, no energy, no transporters. They can be exocytosed, so just kind of spit out through the membrane, if you will. So a piece of that membrane pinches off, we spit them out into the blood, and then from the blood, they can cross this, they're not gonna go into the blood, excuse me, they're gonna cross the epithelial layer here, and they're gonna go into the lacteal, right, which is the lymphatic duct where we can send these off to the general circulation. They're eventually going to go to the liver where they're gonna be sorted, packaged, processed, and then they can be stored if needed. Okay, so this is how we absorb fats, right? Really important to understand fats don't need any transporters or any energy to get into the cell or to get across the basolateral surface. Also important to understand that fats must be packaged as chylomicrons, and then they enter the lacteal, which is that central structure that I would have showed you in the middle of the villi, and then they can go to the lymphatic circulation and then head to the general